Hello, and a very warm welcome everyone to our today's webinar on Phase 1 studies for oncology products. I am Yuvragi Chandeli, and I represent Veeda Clinical Research. Veeda Clinical Research, together with its subsidiary BioNeeds India Private Limited and its joint venture Ingenuity Biosciences, offers a comprehensive portfolio of clinical, preclinical, and bioanalytical services to support innovator, biosimilar, and generic drug development programs of our global clientele. Together, we serve our clients globally in pharmaceutical, biopharmaceutical, agrochemical, industrial chemicals, herbal, nutraceuticals, and medical devices industry. As your facilitator today, I would like to introduce you all to a couple of points as we begin this session. The session will have a presentation in the beginning, followed by a panel discussion at the end. We have enabled our Ask a Question feature. It's right on your screen where you can drop all your questions related to this webinar. Now, moving on, I would like to introduce you all to our guest speaker for today's session, Dr. Vikram Gota. Dr. Vikram is currently a professor at the Advanced Center for Treatment, Research and Education in Cancer at Tata Memorial Center, Mumbai, in the Department of Clinical Pharmacology. He earned his MD from Christian Medical College, Vellore, India, and went on to join the Indo-Oxford Cancer Trials Network at the TMC, where he trained in early phase clinical trials. Simultaneously, he completed the PG diploma in clinical trials from the London School of Hygiene and Tropical Medicine. His department undertakes clinical pharmacokinetic studies, therapeutic drug monitoring, and phase 1 clinical trials of anti-cancer drugs. One of the focused areas of his research include the development of radioprotectors in collaboration with scientists from Bhabha Atomic Research Center. Our second panelist for today's webinar is Dr. Mathak. Dr. Mathak comes with a vast experience of nearly 40 years in the clinical research field. He has conducted many phase 1 studies, including first-in-human studies, complex clinical trials, and more. He has held senior positions in several Indian and global pharmaceutical companies and in leading CROs. And he has vast experience in conducting clinical trials across several geographies, including USA, UK, Europe, China, Japan, and South Africa. Dr. Mathak is highly respected amongst regulatory authorities for his knowledge of international clinical research guidelines. He's also one of the accredited members for GCP training by CDSCA India, and he contributed immensely to the new clinical trial rules implemented in March 2019. Dr. Mathak is also the co-chairman of the Medical Committee of IDMA and a member of the Pharmaceutical and Biotech Committees of the Confederation of Indian Industries. He's a renowned speaker in international and national conferences, besides being actively involved in academics. So without any further delay, I'd like to virtually call upon Dr. Mathak to begin this session. Over to you, Dr. Mathak.
Right. Thank you, Yuvragi, and hello, Dr. Vikram. It's a pleasure to have Dr. Vikram with us today, one of the renowned clinical pharmacologists, particularly for early phase clinical development programs in oncology molecules. So welcome, Dr. Vikram. And with that, I will start my presentation. What I intend to cover is to set up the stage for Dr. Vikram to take over. So I will be talking more on the general aspect on the phase 1 studies. As you know, phase 1 clinical trial is the first in human being to start with. And then there are many other studies that can be carried out, including the SAD study, MAD study, drug-drug interaction studies, gender study, formulation study, food effect study, and also something like renal impaired subject study and the hepatic impaired subject study. So there are a number of studies that can be considered under phase 1 clinical trial. But by any means, the main aim of the phase 1 clinical trial is to find the base dose of a new drug which will have minimal side effect. So that means it has to have some effect on the disease for which it is being produced, and it has to be safe. And is in the phase 1 clinical trial, it's usually carried out in the 30 to 40 subjects, which is divided into 4 or 5 cohorts. And it usually begins with the very low dose of the drug and if it is in a particular cohort. And then slowly the dose is increased to the subsequent doses, the subsequent cohorts. And that is how they reach to the highest dose, and also to that is highest administered dose, and also to learn about the maximum tolerated dose in the group of subjects. And so the phase 1 trial are to be tested mainly as a safety, but certain time it can also give you an idea about a proof of principle or a proof of concept study. However, it also paves the way for the dose for phase 2 clinical trials. Now, the main objectives of conducting the phase 1 clinical studies is to get involved into one or two or more combination of the various parameters. And but main aim is to have a safety and tolerability of a new chemical entity, and to determine the tolerability of the dose range expected to be needed for later clinical studies, as I mentioned earlier, and to determine the nature of adverse reactions that can be expected when the drug is administered in the human being based on the preclinical tests which have been carried out in understanding the new chemical entity. So the main objective is to have some pharmacokinetic activities, which is ADME, that's absorption, distribution, metabolism, and excretion. In phase 1, you can also carry out certain pharmacodynamic activity, and therefore you can do PK/PD studies, which will help in understanding the exact nature of the product as such. And it will also help even in if you are doing a study in patients, like Dr. Vikram will speak very shortly, you can do in PK/PD in a patient population, and will help in understanding the effect of the drug on the disease and how the disease affects the drug. And the early measurement of the drug activity, the disciplinary studies of activity or potential therapeutic benefit that it can provide is a secondary objective. So if we summarize on the objectives of the phase 1 clinical studies, it can provide the safety and the tolerability of the molecule in a studied population. The pharmacokinetics activities, what is the Cmax, Tmax, AUC, etc. We'll see a little more on that in the subsequent slides and how it helps in understanding the molecule. Organs and system susceptibilities, that is on which organ the effect will be, and which organ there can be the adverse events that can be noticed over a period of time. So it gives an idea about the organ and system susceptibilities. It also gives an idea about the metabolism of the drug, how the drug is absorbed, how it is distributed, how it is metabolized, so that you will understand the actual use of the drug. And it can also help in finding out the exact dose which requires for the administration for a particular disease. Estimate the pharmacological activities. Now, this is very important because we have, before one takes up the phase 1 study in animal studies, the studies have been carried out to understand the mechanism of action, but that also needs to be proved in human beings because the species-wise variation could be present. And so it also sometimes also gives an idea about what is the estimated pharmacological activity by doing the proof of concept study. As I mentioned earlier, maximum tolerated dose and the recommendation for the study as such. Now, all this can be achieved in the beginning is in the SAD dose, that is single ascending dose and multiple ascending dose study. In single ascending dose study, the minimal dose which is selected and is tried to in a limited number of subjects in a particular cohort. And in each cohort, there is the one does the PK parameters and also tries to understand the safety profile of the product. And then one moves to the higher second or the next higher dose. And that is how one understands the PK as well as the safety of a molecule. And if one reaches the maximum tolerated dose, that is the observing the expected level of the PK, or the drug produces the serious adverse events or the severe adverse events by which one needs to stop the study. So it gives an idea about, by administering a single dose, what is the safety profile of a product. After doing the single ascending dose study, one goes for the multiple ascending dose study based on the single ascending dose study results, because it will give you an idea about the Cmax, AUC, Tmax, also the half-life. And based on that parameters, one takes over the conduct of the multiple ascending dose studies, starting again with the low dose and going to the higher dose. And the blood samples or the other body fluids, the samples are collected and then analyzed to understand the exact ADME profile of a product. And certain time, it can also give you an idea about the PK/PD and the safety of the drug. Now, all this, when you start the first-in-man study or the first phase 1 study, there is involvement of the multiple groups in the organization, and profile of the product, which helps you in understanding the MRSD, that is the maximum required safety dose in the human being. And then based on that, one calculates the minimum dose or the maximum dose that can be given to start with in the individual. So the toxicologist is very important. And based on the toxicological results and the pre-IND studies which are carried out, the other departments involved are the regulatory affairs specialists, because he has to decide what are the documents required for IND submission or IMPD or CTS submissions required for it, and plan out the strategy for how the study needs to be carried out. There has to be a safety physician who draws up the safety monitoring plan. And one of the very important part in this phase 1 study is the formation of the DSMB, that is Data Safety Monitoring Board. And they have to give green signal after every cohort that has been tried, every cohort that completes the study. And based on the safety profile of that particular molecule at a particular dose in that cohort, one can move to the next cohort with a higher dose, provided it is safe and it is certified by the DSMB. And all this is very important, again, when there is a formulation scientist involved, because he has to ultimately decide what kind of formulation will be required for testing in the human being, whether it is a syrup or oral formulation, etc., based on the product profile as such. And of course, the clinical development scientist, he has to, based on the disease for which the drug is being tried out, he has to develop the clinical development program. And every stage there can be a go, no-go situation. And he has to prepare the whole clinical development program based on that. And also there is a clinical operational scientist who has to select the CROs, where the study is to be carried out, if it is in patients, which sites to be selected, training of the people, etc., etc. And of course, last but not the least important is the clinical pharmacologist, who designs the study protocol. He works out the plan for PK/PD. He also works out the plan for the analysis of the NC molecule in the various body fluids. And if at all, there are certain time biomarkers which plays a very important role in the demonstration of safety and efficacy of the product. Also, the clinical pharmacologist can indicate which kind of biomarkers should be used for the assessment of safety and efficacy. So you can understand that there are many cross-functional collaborations are required for the efficient and successful completion of first-in-human studies. And it also requires sponsors to take a task through a consideration and planning across many disciplines, which needs to be seamlessly integrated. And the first-in-human study should ensure the study design takes into consideration all the preclinical learnings on the drug candidate. So preclinical tests which are carried out, one has to understand and one has to digest the results of it and make the best use of it when one designs the protocol for the first-in-human study. So in designing the study, there are multiple factors which one has to keep in mind. It's not only SAD and MAD study, but also how many subjects, what is the study population, the dose escalation scheme, specification of dose limiting toxicities. If there is the study is being carried out in patients, then what kind of patients required, and also what will be the secondary objectives, the primary endpoints, secondary endpoints, everything needs to be calculated and to be put down in the protocol as such. And sponsors may incorporate many times in one protocol multiple aspects like multiple dosing, food effect study, relative bioavailability study, QTC prolongation study, or even for a particular molecule. There is also a tendency to use umbrella protocol, which allows an adaptive trial design in which the data is made available. One has to study that data when the study is still going on, and it permits you or it allows you to make slight modification in the protocol based on the result that one has seen, and it helps in understanding the product in a much better way. And in that case, one can change certain parts in the protocol, maybe the dose, the duration, the kind of population, etc., or even certain biomarkers, the safety profile, all these, so that one does not administer the drug into a wrong population or does not administer the wrong dose. So this umbrella protocol is becoming very common now, particularly in the phase 1 studies. As I was mentioning earlier, there is pharmacokinetics is one of the very important aspect in the phase 1 study. And it gives you an idea about because you're trying the various dosages in the different cohorts in the ascending dose parameter. And it can give you an idea about whether it is dose proportional, the PK profile is dose proportional, or whether it is greater than dose proportional, or whether it is lesser than the dose proportional. And each one, if it is more or if it is low, it has its own drawbacks. And there are various studies that needs to be planned. One has to be very careful in increasing the dosage. It also gives you a narrow margin of the activity, including the safety. So this PK is very important at the various dosages, and the dose proportionality is one of the very important aspects to be considered in the phase 1 study, particularly in the first-in-human studies. The other part that one has to keep in mind is the effect of the food on the drug as such. And you can see this profile here, the two curves that I have put here. The one in the red is the one which is on fasting, the PK parameters, and this is with an NCE, one of the study that I have conducted. And when it was administered in the same subjects, but after food, there is a tremendous increase in Cmax and AUC. And this increase is at least 4 times increase in Cmax and 5 times increase in AUC. And that helps in lowering the dose. And when you administer the drug in the patient or in the subjects after food. So the advantage would be that one can administer the drug at a lower dose and one can get the lower side adverse events profile also. So food effect study is one of the very important studies that needs to be carried out in phase 1. Also, there are different formulations, as you know. There can be then immediate release formulation. There can be the prolonged release formulation, and there can be a delayed release formulation. And these formulations, ultimately the formulation scientist decides based on the initial PK parameters what needs to be done, whether one should prepare a modified release or delayed release or a prolonged release tablet formulation. So this is again also, and this has a tremendous impact on the adverse event profile of the product. I also mentioned about the gender effect. I did one study in which you can see this is for the male subjects and this is for the female subjects. And in female subjects, the PK parameters or the Cmax was nearly 50% higher than compared to the male subjects, and including the AUC also was on the higher side. The advantage was that in the certain indications, which is mainly for female subjects, we could reduce the dose. Instead of giving 100 milligram dose, we could give 50 milligram. We changed the protocol and we could see the results which we could have seen with the 100 milligram with 50 milligram tablets. And this also again reduces the adverse events profile. So all those PK parameters, those examples, just I wanted to draw your attention so that you can understand the value of doing the phase 1 and value of PK profile and the various phase 1 studies. Now, maximum recommended starting dose, Dr. Vikram will take it more in detail, but one has to keep in mind that it is based on the preclinical studies which I carried out, particularly NOEL, based on the animal data. And one has to keep in mind that the initial clinical dose should be such that it should not produce the adverse events. And there are different ways of calculating it, which Dr. Vikram will cover more in detail. Also, you can carry out the immunogenicity testing, particularly when you are dealing with the biotech products. And in these cases, then it gives you an idea about the immunogenicity profile of a product and also gives you an idea about the neutralizing antibodies. Only thing is one has to keep in mind that the study population should be as per the requirement, and that one has to keep very much in mind, and then you can understand the safety and efficacy of a particular product. Phase 1, because we are doing it for the first time in human being, there are very safety parameters one has to keep in mind. And those safety factors are, one of the safety factors is the extrapolating the animal toxicity studies in the human being. And that could be very tricky because certain time there is uncertainties due to enhanced sensitivity in humans versus animals. There are certain adverse events like headache or myalgia, the animals will not complain. There are certain kind of receptors which have got more densities in human beings, or certain receptors densities are more in animals. So it depends on which kind of receptors we are looking at. There could be unexpected toxicities that can happen, that some adverse events, toxic profile which was not observed in human, but not observed in animals, but it was observed in the human beings. It can happen. And the interspecies differences in the ADME profile of the product. So this could be a variability, and one has to therefore keep very close vigilance while conducting the phase 1 study, and particularly the first in human being studies. There can be a steep dose response curve, which may indicate a greater risk in the human beings. There can be a non-linear pharmacokinetics, as you could see in the earlier graphs. It limits the ability to predict the dose reliability and the dose-related toxicity. And if there is variability in the bioavailability, then particularly if there is a poorly bioavailable product, then it can underestimate the toxicities in human beings. So safety is very important in phase 1 clinical trials. And the overall incidence of serious adverse events is roughly about 0.02% in the either in healthy subjects or the patients. Now, all this study, it can be either carried out in the healthy subjects or in the patients. It can be carried out in healthy subjects, particularly when one does not want to have any comorbidities or concomitant medications coming in the way of interpretation of the clinical results, or even interpretation of the PK parameters, or even the safety of the product. So in such circumstances, the the healthy subjects are selected. But there are certain products which have got an inherent risk of drug candidate, which cannot be tried in healthy subjects because they are either cytotoxic as such the product, or it is in the targeted for the life-threatening conditions where there is no other therapeutic options available for the patients, and therefore one can try out in the patients to give them hopefully giving them some benefit of the product as such. And so or even the invasive dosing route is required for the dose administration. So it can be done either in healthy subjects or in patients. And as I mentioned here, most of the cytotoxic drugs, the phase 1 study, the first in man study, is always carried out in the patients as such. And with that, I hand over to Dr. Vikram, who is specialized in conducting phase 1 studies in the oncology patients. Over to you, Dr. Vikram. Thank you very much.
Thank you very much, Dr. Marthak, for laying the general considerations for conducting phase 1 clinical trials. And now, as you rightly mentioned, I'll be talking about the requirements for conducting, trial patients. I think, there is some issue here. 1 second. So I'll just start sharing my slide deck. I hope my slides are seen.
Yeah.
Okay. Yeah. So as I said, you very beautifully illustrated the simple designs that are used in healthy phase 1 clinical trials, and also you touched upon certain operational aspects that are very, very important when we move into understanding phase 1 clinical trials in oncology. So the mandate for me for this evening is to discuss about phase 1 trials of oncology products, and that is what I'll be doing in the next half an hour or so. So just to provide a brief outline of my talk, I'll be talking about phase 1 clinical trials in oncology, and then the IDEAC recommendations, which is the Investigational Drug Steering Committee recommendations for designing phase 1 clinical trials. I'll touch upon dose escalation designs, and also the Bayesian designs to end my talk. So phase 1 clinical trials. First, human trials of experimental or approved drugs in novel combinations are generally called as phase 1 clinical trials. So earlier, the phase 1 were a very simple design, as Dr. Marthak rightly pointed out, wherein you kind of escalate the dose from a very low level to a very high level till the patients or volunteers had some discernible toxicities, what you would call as dose-limiting toxicities. And based on that, you would identify a safe dose to be taken for phase 2 clinical development. But from there, all the way to the present day, where we can actually match the patients for drugs. Like, for example, he alluded to basket trials and umbrella trials. The phase 1 trials have made huge strides. And in fact, we are having more and more model-informed drug development, which I'll be talking about in due course, which has actually changed the facade of phase 1 clinical trials. And therefore, it is a very interesting space in drug development in the present day and age, with so much of mathematics and statistics coming into play. Okay, so some of the important therapy areas for which phase 1 trials are kind of ongoing at this point in time are cancer, digestive diseases, and endocrine and metabolic disorders. And this graph here shows how the phase 1 clinical trials have, the number of trials have increased in the last decade. And this is data from China. And of course, I can tell with certainty that the same trend is kind of present across the globe. So now, phase 1 acts as a bridge between preclinical development and late phase clinical trials. So we have the preclinical data coming from mice and monkeys and so on. And then we have late phase clinical trials where people are looking for efficacy and effectiveness of drugs. And then in between there is a void which is filled by phase 1 clinical trials, wherein you will try to estimate what happens if I give a drug. How does the pharmacokinetics look like? What are the conditions under which the pharmacokinetics can change? As Dr. Martha rightly pointed out about gender and age and so on and so forth. So food effect and so on. So pharmacokinetics, and then it will help us to identify. Okay, I have to conduct phase 2 and phase 3 clinical trials going forward, but what dose do I choose? Okay, and how do I select that dose? So these are some of the important considerations that are actually dealt during phase 1 clinical trial. Okay, now coming to IDEAC recommendations for designing phase 1 clinical trials, and why are IDEAC recommendations important for us? That is because IDEAC works in close collaboration with National Cancer Institute USA, and then they make recommendations for phase 1 clinical trials. And if we have some kind of a general understanding about what these recommendations are, we are less likely to make mistakes in design and the escalation schemes and starting doses and so on and so forth. So therefore, the recommendations are made under 3 large headings. So first is the trial design. So IDEAC says any trial design is acceptable as long as it answers the question of interest within the ethical parameters of conducting clinical trials. So 3+3, it could be a 3+3 design, it could be an accelerated iteration design, it could be a Bayesian-based design, whatever. Okay? But the only recommendation that they want to make in terms of selecting the design is that we have to reduce patient exposure to non-therapeutic doses. Because that has been the bane of clinical trials, phase 1 trials in the past, where a number of patients would be exposed to subtherapeutic doses before we could move to the business end of the dose. So this is one area which IDEAC has recognized as a problem area, and they have made these recommendations. Then what are the endpoints that you should have for a phase 1 clinical trial? Toxicity, obviously, because this is the first time the molecule is being tried in a human being, and we don't know what to expect from the drug. So safety first is the approach. So therefore, toxicity definitely. And Dr. Martha has already spoken about the maximum tolerated dose, but I also talk to you about the recommended phase 2 dose and the biologically active dose. So MTD is a very interesting concept, in phase 1 trials. So what MTD means is if I dose patients at a certain dose, of the drug. So what is the highest dose that I can give, wherein not more than 1 out of 6 patients will have dose-limiting toxicity. Okay, so that defines the safety boundary. Okay, so not more than 1 out of 6 patients can have a dose-limiting toxicity. And what is the dose-limiting toxicity? I can talk for oncology trials because that is what I generally do. Dose-limiting toxicities are generally, you have to define it for every clinical trial. And it can differ by slightly from trial to trial. But typically, definition of a dose-limiting toxicity is any toxicity which is grade 3 or higher in nature when it comes to the non-hematological systems. And for hematological toxicity, generally grade 4 toxicities are considered dose-limiting toxicity. So now, having understood DLT, if more than 1 out of 6 patients experience a dose-limiting toxicity, that dose is called as the maximum tolerated dose. And of course, there is slight difference in the interpretation of MTD across the Atlantic. The Europeans have a slightly different definition. The Americans have a slightly different definition. We can discuss that in due course. Okay. And then the committee also recommends that you should consider intra-patient dose escalation wherever possible. It is reasonable because it will avoid patients being dosed at subtherapeutic doses. Okay. So now the third set of recommendations are related to combination treatments, and I'm not going to get into combination treatments because I want to keep this talk simple. In half an hour, we can only discuss so much. But suffice to understand that when we're talking about combination, there should be a sound preclinical or mechanistic rationale for launching a combination phase 1 clinical trial. Okay. So clinical trials of different drugs, similarities and dissimilarities. So here the drugs are the cytotoxic agents and the targeted agents. So MTA stands for molecular targeted agents or simply targeted agents. Okay. So what are the differences? Now, the endpoint of interest for both these set of compounds is RP2D. One must understand that we are slightly moving away from MTD to a concept called RP2D, because MTD is defined only by toxicity, whereas RP2D is defined by a combination of toxicity and then pharmacokinetics, pharmacodynamics, efficacy, and so on and so forth. So therefore, RP2D is being considered more and more as a reliable definition of a dose, to be considered for phase 2 testing. So therefore, and also there is a problem with MTD. The way MTD is calculated, in oncology, at least I can tell you that, it is generally for the first 28 days of observation, because most cycles in oncology are 28-day cycles, because chemotherapy or any drug is given cyclically. So historically, what has been happening is MTD is observation of toxicities or DLTs for the first 28 days. But one must understand that, toxicities can happen beyond the 28-day period also, because of accumulation of drugs, or simply because of the multiple doses that the patient might receive 6 months into treatment or 12 months into treatment or so. So therefore, just going depending on the MTD for phase 2 dose identification is not a good strategy. So therefore, more and more people of late are using what is called as the recommended phase 2 dose. And it is also important to understand that the RP2D is generally lower than the MTD. It may be either the same as the MTD or one step lower, but it is definitely never going to be higher than MTD. So the secondary endpoints are toxicity, response rates, and so on for cytotoxic drugs. Whereas for molecular targeted agents, we are more worried about the PKPD concentrations. Of course, toxicity will continue to be a matter of concern, no matter what type of drug it is. The dose escalation decisions are based solely on toxicity when it comes to cytotoxic drugs. But here, because the secondary, if you look at the primary and secondary endpoints, it becomes obvious that it is not just toxicity, but it's also about the PD outcomes that becomes important when you consider dose escalation. And PK parameters are calculated in both scenarios. And the basis for selecting RP2D is toxicity once again for cytotoxic agents, but it is a combination of toxicity, PKPD, and activity and tolerability, all taken together in the estimation of RP2D in the case of targeted agents. Now, the concept of maximum recommended starting dose is very, very important. And Dr. Marthak finished his talk with MRSD. So now, what is MRSD? So let's say, when a drug comes to phase 1 development, hardly anything is known about the drug. At least the human data on the drug is non-existent, because that's why it's called as the first in human study. Okay, so when you don't have any human data, so how do you start? Where do you start from? Is 1 milligram a good dose, a half milligram a good dose, 0.1 milligram a good dose, or should you start with 100 milligrams? And mind you, this starting doses is different for different agents, based on the safety profile and based on the receptor occupancy, based on the potency, and so on and so forth. So therefore, there are two approaches primarily to identify what the maximum recommended starting dose. One of them is called as a Noel-based approach. Noel stands for no observed adverse effect level. It's a very straightforward thing. What you do is, you identify what is the dose, highest dose, which causes no toxicity in animals, and then you calculate what is called as a human equivalent dose, and you add a safety factor to that. So let us say the highest dose was 50 milligram per kg that did not show any toxicity in the rat. So you apply, you calculate the human equivalent dose, and once you calculate the human equivalent dose of the mouse dose, you add a safety factor, and you have the starting dose. But now that will work very well for cytotoxic drugs. But for targeted agents and more recently for immunotherapies and so on, that is not a very good estimate of the starting dose. So therefore, more and more we are relying on what is called as the minimum anticipated biological effect level, which also takes pharmacology into consideration. Okay, so therefore it's not just toxicity, but it is also pharmacology based. I'll talk to you about this one in the next couple of slides. So it's suffice to know that the maximum recommended starting dose, it should be a safe starting dose in humans. Okay, it should be a safe starting dose in humans, and I'll talk to you a little more about this in the next couple of slides. So now, when I spoke about identifying the human equivalent dose, I said, you have a mouse dose of 50 milligram per kg, which is identified as Noel. How do you identify the human equivalent dose? So that is done using this conversion table. So basically, when you know what is the dose in a given species, let's say mouse, okay, if you have to convert to the human dose, you have to multiply the mouse dose by this Km which is the mouse km, and divided by the human km. So if the dose is 50 milligram per kg in mouse, you multiply 50 into 3 and divide it by 37. So essentially, you will divide the number by 12, which will give you the human equivalent dose. And so, likewise, if the safe dose is found to be 50 milligram per kg in monkeys, you please remember this number because I'm going to talk to you about this a little while later. So if the safe dose is found to be 50 milligram per kg in monkeys, what you do is you multiply 50 with 12 and divide by 37, and that will give you the human equivalent dose. So it is essentially 1/3. 12 upon 37 is 1/3, more or less. So the human equivalent dose on a kg body weight basis is essentially 1/3 of the monkey dose. Okay. Now this graphic is very, very important for the understanding of maximum recommended starting dose. So what do we have here is a dose response curve in animals. Okay, the solid lines depict the dose response curve in animals. Okay, so this is the dose response curve for toxicity, which is at the far right, and this is the dose response curve for pharmacodynamics, which is somewhere in between, as you can see here. So now from this you will kind of extrapolate and kind of calculate or extrapolate the dose response curve for humans for these two parameters. Dose response curve for toxicity, which is the third line from left, and dose response curve for pharmacodynamics, which is the first line from left. Okay. So now, how do you calculate the maximum recommended safe dose? So one is a novel-based approach, which is very simple. You calculate the novel. Okay. You calculate the human equivalent dose. That is the step 2 and 3. And once you calculate the human equivalent dose, you add some kind of a safety factor, which is generally 10 or 20 or 50 or whatever. And then you will get what is called as MRSD star. Okay, just remember this, MRSD star. And on the other hand, you also have done this extrapolation, and then you would have identified what is the minimum. Can you see this point here where I'm pointing to? So you would have identified what is called as the minimum anticipated biological effective dose, which is nothing but the minimal pharmacodynamic effect. So that will be your Mabel-based MRSD. And whichever is less between MRSD and MRSD star, that dose can be considered as the first in human dose or the maximum recommended safe dose. Okay, so you please remember this graphic, and I encourage all of you to read this reference from Muller et al., which depicts this concept very, very nicely. Okay, so now what are the considerations for finding the starting dose for molecular targeted therapies and also the dose escalation? So it is very simple. Targeted agents, they may not produce the same kind of toxicity that you find with cytotoxic drugs. So therefore, sometimes you may not achieve the maximum tolerated dose at all. So you go on escalating, but you don't find any dose-limiting toxicity. So then you have to settle for what is called as the maximum administered dose. Okay, so that is about the dose escalation. Now, how do we calculate the first in human dose? Like for example, because they are very safe, what you do is you will calculate what is called as the MELD10. MELD10 basically stands for 1/10 of the murine equivalent lethal dose in 10% of animals. So, like for example, if you have dosed 10 animals at a certain dose, and if it causes 1 death, okay, so that is the MELD10. So from that, you calculate the human equivalent dose, and from there you have a safety factor of 1 upon 10, 10 times lower dose, and that will give you the starting dose. Or if you're using a non-rodent data, because non-rodents like non-human primates like monkeys, they are much closer to humans in terms of safety and efficacy effect, right? Then you can use that one. I mean, in the sense, the dose obtained from non-humans as the starting dose, and then when you're escalating the dose, you can usually go for a doubling of the dose. Okay, so there are two strategies for dose escalation. One is a more conservative strategy, which is given by the Fibonacci sequence, wherein you start with 100 or X, then you go to 2X, then you go to 3X. The Fibonacci sequence says each subsequent number is sum of the previous two numbers. So you start with X, 2X, 3X, because 3 is equal to 1 plus 2, and then 5X, because 5 is equal to 2 plus 3, and so on. Okay, so there is a very conservative dose escalation defined. But if you want, or if you think that it is too conservative, you can settle for what is called as a modified Fibonacci, wherein you can go in incremental ratios like, you double it, then, the next dose will be 1.67 times, 1.5 times, and so on and so forth. So these are the recommendations if you want to play very safe, depending on the nature of the drug. But if the drug has a low toxicity as you find in animal models, and if you think that it's going to be very safe in humans, you can easily go for doubling of the doses. Okay, so now what are the various methods that are used to estimate the first in human dose? We already discussed about novel. We already discussed about Mabel. Okay, so the novel advantage is that it has stood the test of time. It has stood the test of time for several decades. A novel-based approach has been in use, so we know how it works, and it has kind of worked reasonably well. A Mabel-based approach is definitely required for targeted therapies, and more recently for immunotherapies. Definitely, you should use a Mabel-based approach, and I'll tell you why in the subsequent slides. But only problem is it requires extensive non-clinical data because you are going to decide the pharmacological, the minimally anticipated pharmacological dose. So therefore you should need extensive non-clinical data, not only of toxicity but also of efficacy in animal models. And it is also unclear which species will correlate better. Like for example, whether you should use mouse data or whether you should use rat data for estimation of Mabel is not very clear sometimes. So that can be a problem. For PK model-based identification of first in human dose, it has some advantages. Like it accounts for species differences in PK aspects. If you know the chemical nature of a drug, you can pretty much anticipate what kind of differences are going to be there between humans and the species of interest. And so therefore you will be able to calculate the safety margin, and it works well for drugs which have linear pharmacokinetics. So linear pharmacokinetics, you can easily do a PK-based identification of first in human dose. But there are certain assumptions which may or may not hold all the time, and the assumptions are like, the dose effect relationship. It assumes that there is a different dose. Like for example, the same concentration which is effective or safe in animals, it is assumed that the same concentration will also be effective or safe in humans. So that may or may not always hold. So that is one of the limitations of using a PK-based approach for identifying the first in human dose. A more nuanced approach for identifying the first in human dose is based on PKPD modeling because this is more mathematical in nature. So when you bring in mathematics, you will your estimates are slightly better. You will build some confidence intervals around your estimates, and then with experience you can always make those intervals smaller and more precise and so on and so forth. So therefore, a PKPD modeling based approach is much more reliable. But the only problem is it requires an experimental experienced model development team. So which is good in quantitative pharmacology. So just to give you an overview, these are the 4 different approaches that are available for identifying the first in human dose. But the most commonly used ones are the novel-based approach, which is used for almost all cytotoxic, and the Mabel-based approach, which is used for targeted agents as well as immunotherapies. Okay, now how does the dose escalation designs work? This is a very simple 3+3 design. So the 3+3, I think I'll talk about the 3+3 design in the next slide because it is more clear. So basically, you escalate till you find the dose-limiting toxicity. Wherever you find dose-limiting toxicity, you put 3 more patients. That is called 3+3 design. And if you have any scenario where at a dose where more than 1 out of 6 patients have a dose-limiting toxicity, that is called as the maximum tolerated dose in US, or in Europe rather, or 1 dose below that is called as the maximum tolerated dose in USA. So basically, in this scenario, you are going to take this dose level for the next phase, that is phase 2 clinical trial, because here only 1 patient out of 6 had a dose-limiting toxicity, but as we went to the next higher dose, 2 out of 3 had dose-limiting toxicity. So obviously, it violates the safety margin. But a variation of this one is, so if you want to avoid dosing too many patients at a very low dose, you can use what is called as the accelerated titration design, wherein instead of putting 3 patients, you can just put 1 patient at any dose. And suppose at any given dose, you observe toxicity which is not amounting to DLT, but there is some toxicity, then you kind of change over from an accelerated design to a classical 3+3 design, and then you identify your maximum tolerated dose according to that. So this is a slight variation of the 3+3 design, which is called as the accelerated titration dose. design. And the ATD also allows intra-patient dose escalation as we discussed previously. So therefore, sometimes some people find it a little more efficient in terms of coming to the business end of the dose very, very quickly as compared to the standard 3+3 design. Once again, I'm reiterating the standard 3+3 design. So wherein you put 3 patients and then none of them have a dose limiting toxicity, you escalate. If one of them have a dose limiting toxicity, you put 3 more patients. If only 1 out of 6 have a dose limiting toxicity, you can once again escalate. If more than 1 out of 6 have a dose limiting toxicity, you have to stop the drug. And here, if 2 out of 3 has a dose limiting toxicity, you have to stop the drug and you have to consider the previous dose of the maximum tolerated dose. Now, coming to the MTD definition itself, I told you before, in Europe and America, across Atlantic, there is slight difference in the way MTD is defined. In America, generally the one dose level below the dose that causes more than 1 out of 6 patients with DLT is considered the maximum tolerated dose. But in Europe, it is that dose which causes toxicity in more than 1 out of 6 patients is considered the maximum tolerated dose. It is just semantics. There is no difference in science. The science is always the same because the dose level that you take to the next phase is always the same, whether you are in US or America. It does not change. Okay. So now this is a very interesting story that I want to narrate. So how did this 3+3 design originate? Okay. So this the 3+3 design was first used in explosives. Okay, in testing explosives, and it was not originally designed for phase 1 clinical trials. So how does it work? So you have an explosive and you want to test test an explosive. Okay. So you provide a stimulus, which is like some kind of a fire or whatever, and then you have some loading of the explosive inside this cartridge. Okay, and then you fire it. Okay, and then you look at the response. If the response is present, you kind of so you define what should be the decibel of the explosion, or what is the damage that is created with the explosion that you define. And then if it is more than that, you decrease it. If it is less than that, you increase the stimulus. So, okay. So if it is more than that, if it's positive, then you decrease the step size. So you decrease the amount of let's say dynamite put inside the cartridge. Or if it is negative response, then you increase the stimulus. So now an analogy to that is your sample of explosive are the patients. Your stimulus is nothing but the drug treatment, and then your response or non-response is the DLT. Like for example, if there is a response, it can be considered as a dose-limiting toxicity, and then eventually what you will do is you'll identify the right dose to be taken forward. So this is how the analogy stands. But one must understand that the 3+3 design itself came for testing the explosives and not so much with the phase 1 trials in mind. Now, you might be wondering why 3+3? Why not 1+1 or 2+2 or 3+3? No, by definition, see, before we go to this point, we must understand what is called as a target toxicity level. A target toxicity level is defined as the dose at which, which not more than, let's say, 1/3 of patients develop dose-limiting toxicity. So the dose range is from 1/5 to 1/3. That is 20% to 33%. Okay, so that is called as the target toxicity level. So any dose which gives you DLT in the range of 20 to 33% is okay for us. If the DLT exceeds 33%, it is not okay for us. Okay, so therefore, that is how the target toxicity level is defined. Now, if you want to define the target toxicity level, obviously you cannot define it 1 or 3 patients because 1 or 2 patients, because at least 1/3 means at least you need 3 patients, so that you can define that 1/3 of patients are dose-limiting toxicity. So therefore, 1 or 2 patients is out of the window. Now, why not more? Why not have 6 or why not have 12? So I'll just give you one example of why we cannot have more number of patients at the dose level. And this came from the 2006 experience of the CD28 super agonist antibody, which was conducted in the UK. So what happened was, they enrolled 5 patients at each. The idea was to enroll 5 patients at each dose level, 5 or 6 patients. So the dose, the first in human dose was calculated in a very conservative fashion. So actually, the monkey dose which was shown to be safe, was 50 milligram per kg. So what they did was they used a novel-based approach and then calculated the human equivalent dose, which came to, I mentioned earlier that with the monkey dose, you just simply divide by 3, it came to 16 milligram per kg. And then they further added a safety factor of 160. So they used 160 times less than the human equivalent dose, and they started with 0.1 milligram per kg. But what happened was, after the first infusion, there was a severe cytokine storm in all the patients, and all the 6 patients, they developed multi-organ failure and resulting in death. So therefore, what is the lesson we learned from this is human trials of potent biological molecules should include initial testing on very less number of human volunteers before administration of drug to a greater number of human volunteers, just so that you minimize this kind of a situation. Okay. So now I'll move forward with 3+3 design. So there's a very simple calculator that is available for calculating the number of the cohorts and the dose that you have to dose, the dose that you have to use at each cohort and so on. So it actually requires 2 inputs from you. So you first have to select whether which type of study you want to do. Where you want to do 3+3 design with the which allows dose de-escalation or which does not allow dose de-escalation. Okay, so this is the first decision you have to make, and then you have to also enter what is the probability of dose limiting toxicity at each of those doses. Like for example, here it is entered. Like the probability of dose limiting toxicity is 5%, 10%, 15% at each of the doses. You calculate the first in human dose based on the criteria that I discussed earlier. Like you can use a Mabel approach or a Novel approach or whatever. And then you just tell how many cohorts you want to use. And once you give these inputs, it will give you the various doses that you have to try, and then you are set to go ahead with the classical 3+3 design. Now the other types of designs, as I mentioned earlier, are the pharmacokinetically guided dose escalation. I'll come to a cartoon in the next slide, which will tell you how the PK guided dose escalation actually works. Okay, the only problem with that is it requires real-time PK assessment for dose modification, and it also assumes that DLT can be predicted by plasma drug concentration. Okay, so then there are the more complex model-based designs, which I'll touch upon more in the later slides. And there are several variations of the model-based designs, like you have escalation with overdose control, continuous reassessment method, modified CRM, tentative event CRM, and so on and so forth. Okay, so now let me talk about the pharmacokinetically guided dose escalation design. This is nothing but a classical 3+3. Okay, so or rather, you can say it is more like an accelerated iteration design. So you can kind of go to a certain stage. Okay, and then here the legend is very important. Red circle means there is a DLT and the PK threshold is met. Green yellow circle means that there is no DLT, but still the PK threshold is met. And green circle means that there is no DLT and PK threshold is not met. So initially you take only one patient each, and then see whether there is any PK threshold being met. Okay, initial escalation is only for PK threshold, but after a certain stage you become a little cautious. So instead of just escalating with a single patient, you want to get more safety data. So you put 3 patients, and then somewhere here you will find that you have met the PK threshold. The PK threshold is defined from preclinical studies, and that is the assumption that you make. Okay, so I can extrapolate the preclinical PK data to human PK. So you only if you are sure about that assumption can you use a pharmacokinetically guided dose escalation. So you do that. And suppose you have achieved the PK, but you have not reached the dose limiting toxicity. Just like the classical 3+3, you escalate to the next higher dose and see that, okay, obviously in the next higher dose, the chances are that almost all of them will have the PK, threshold achieved. You must understand that, here in the 3rd or 4th level, what is happening is 2 of them have achieved the PK level and 1 of them has not achieved the PK level. That can happen because of the interpatient variable. Okay, so there's another thing you must know. But as you go to higher doses, the chances are that more patients, more proportion of patients will achieve the PK guided PK target. Okay, but suppose there was 1 dose limiting toxicity here, then you put 3 more patients, and then if there are no more DLTs, you escalate, and then if you are at a dose level at which you find more than 1 out of 6 patients having DLT, you define as the maximum tolerant dose. So this is how a PK guided dose escalation works, and of course there are some assumptions as I mentioned, and you should be careful about that. And now coming to the model-based designs, the model-based designs are slightly different. Here, I'll just give you a background. So I told you about the target toxicity level, which is in the range of 20 to 35, which is acceptable. So you must have that understanding. And what is model-based designs? Basically, they use Bayesian methods. So they will define what is called as the prior probability distribution. So prior probability distribution of DLTs. Okay, how is it done? It is done based on your experience with the preclinical models, or you may have some idea about how it is going to be based on the class effect, because you may have a similar class of drug already in the market, so you may have some experience with the drug, and this is a drug from the same class, so you know how the toxicity profile is going to be. So either you have from the preclinical experience, or your knowledge about the same class of drug, or you can make a simple guess, okay, whatever it is, but you define some kind of a prior DLT probability distribution. Okay, so prior DLT probability distribution, and then you start putting patients. So once you start putting patients, you will calculate, suppose I put about 5 or 6 or 7 patients or 3 or 4 patients, I'll calculate from the prior probability distribution of DLT, I'll refine that to what is called as the posterior probability distribution. Why posterior? Because the new distribution of DLT probability is based on your experience with these 5 patients. Okay, so you define what is called as the posterior probability distribution, and then from then on you escalate, because you know what dose is likely to give you the target toxicity level, and you keep on escalating till once again the same rule is satisfied. More than 1 out of 6 patients are get dose limited toxicity, and that is how you define. So here it is more efficient because it is mathematically guided. So you are kind of more likely to be accurate about your estimates of the probability of DLT, and it also allows faster escalation, because you would calculate the probability of DLT and you know at what dose level the DLT is likely to happen. So therefore, instead of going stepwise in a Fibonacci sequence, you can kind of escalate 100%, 100% or 200% or 300%, and so on. So therefore, these designs are likely to be more efficient, and these are also called as CRM, continual reassessment method, because you are continually assessing the toxicity and then defining your posterior probability of DLT. So the main difference between 3+3 and CRM is 3+3 is rule based. You have to strictly have 3 patients at every stage, and then you can go only in one direction. But CRM will allow you to come down also. Like for example, if you overshot your DLT threshold, then you can come down. Okay, and then at any point you can switch from a conservative escalation of let's say 40 or 50% to a 100% escalation based on your posterior probability of DLT. Okay, so therefore it allows you to reach the MTD much quicker. Okay, so now I'll just briefly talk to you about what is called as a model-assisted drug development. Okay, so this is it's a model-based, where CRM is model-based drug phase 1 design, but here this is model-assisted. Basically, this gives you the mix of both a rule-based design and a model-based design. Okay, so rule-based design says what? 3+3 is a rule-based design. So to put 3 patients, then 3 more patients, observe for DLTs, then the DLT you add 3 more patients, and so on and so forth. That is a rule. Okay, so this also follows a similar rule, but this kind of gives you best of both worlds. So at the same time, it also uses some kind of a probability estimate that you would have kind of defined a priori. Okay, here it will only take the prior probability estimates, prior probability distribution. Unlike the CRM, which does the posterior probability distribution, it does not. This design does not do the posterior probability distribution. It only does the prior probability distribution estimates. So therefore, at each target dose level, you would have defined what is the likely toxicity to be, and what is the lambda E. Lambda E means below this, you will calculate the DLT incidence or the DLT rate at each dose. And if the DLT rate is less than the dose escalation boundary, you will escalate the dose. Likewise, if the DLT rate is more than the dose escalation boundary, you will de-escalate. If the DLT rate is between lambda E and lambda D, that is, it is neither below the dose escalation dose de-escalation boundary, then you will kind of continue with the same dose. Sorry, I'll have to shift places because my battery is discharging. Okay. Okay, I hope we're set now. So now, how does it work? You start with certain dose based on either Mabel or Noel or whatever is your preferred method of you treat a cohort of patients, and then the stopping rule is always there. That more than 1 out of 6 patients developing DLT, you have to stop, no matter which design you are using. If that criteria is met, you have to stop. If not, you will compute DLT. How do you compute the DLT rate? Total number of patients with DLT upon total number of patients treated at the current dose. So that will give you, let's say, it will give you a DLT of 15% or 16%. 1 out of 6 developed, let's say. 1 out of 6 developing DLT is 16%. Okay. So then at that dose, you will say, okay, where does 16% lie? Suppose you are in the first dose here. Okay. So 16% lies somewhere here. It is kind of more than your lambda E, but less than your lambda D. So which means that you cannot escalate. Okay, you put a few more patients. Suppose when you do that, your next estimate of the DLT rate came to about 10%, let us say. So then it is below the lambda E. So which means that you can escalate. Then you will prove that the dose is safe and you will escalate to the next higher dose. So this is how it goes, and it is called the optimal interval design. Okay, and here the advantage is it follows the rule-based method also, because everything is defined by prior, so you cannot, it is not flexible. But at the same time, it allows the advantage of using a mathematical model. Okay, so this table actually shows the difference between the various designs: the 3+3, the model-based, the CRM design, and the model-assisted. It's called BOIN. Okay, Bayesian optimal interval design, BOIN. Okay. And then here, the BOIN will behave more or less like 3+3, but still it has some advantages, like accurate MTD estimation, because it is kind of based on some mathematical rationale. Your estimates of MTD is likely to be more accurate. Of course, the CRM offers the most advantages in terms of the various characteristics of design characteristics, like flexibility of the design. So the CRM is flexible. The quant size is flexible in CRM. It is flexible here, but 3+3 is very rigid, as I said. So it is flexible here because every time it comes within that limit of lambda E and lambda D, you have to put more patients at the same dose. And the moment you either exceed, go below lambda E or go above lambda D, you can either escalate or de-escalate. So therefore, the sample size is not fixed in this case, but the sample size is fixed here at every cohort. So therefore, there is no flexibility of cohort size. You cannot exceed 6. But here you can go to any number of patients. So so dose toxicity relationship is best when it comes to CRM, but neither the 3+3 design nor the model assisted design will give you a good dose toxicity relationship, because here the probability of DLT is continuously assessed and the posterior probability is redefined at every step. So these are some of the this is how you can compare and contrast the 3 designs. Okay. So just to kind of give you an overview of whatever we discussed so far, 3+3 designs, the problem is that, you treat more patients at subtherapeutic doses. Okay. So that is the problem. And then the simulations have shown that your estimate of RP2D you kind of estimate the RP2D at lower doses than other designs. Like, for example, if CRM says that RP2D is 100 milligram, you are likely to find RP2D at 80 milligram in a 3+3 design. Accelerated titration design allows intrapatient dose escalation. It may sometimes be a problem because the accumulated toxicity may also contribute to MTD and RP2D. So therefore, that may be a problem. It may not help you to arrive at a correct estimate of the RP2D. Pharmacokinetic dose escalation, it makes some underlying assumptions, like the same kind of exposure effect relationship you have in mice. Let's say you have similar relationship in humans. That may be a flawed assumption. That is a problem. I'll talk to you about the overdose control, escalation design with overdose control in the next slide. But it is a very interesting concept and something which allows you to determine the RP2D with greater accuracy. CRM is definitely good, but the only thing is because it is more ambitious, the dose escalation is ambitious, you may sometimes overestimate the RP2D. Like if the RP2D is, let's say, 50, you may estimate the RP2D as 60. So that can be a problem. And that is overcome using the modified CRM design. I'm not talking to you about the modified CRM at the time to event CRM in the interest of time. But so now my last stretch of my talk is about the Bayesian logistic regression method. So this is definitely, I'm not going into the details of this, but this is definitely a better model. It utilizes all information like the CRM does. Okay, this is nothing but a CRM, but in addition, it has called what is called as it has got what is called as the escalation with overdose control. So I'll talk to you about overdose control. The key difference between CRM and BLRM Bayesian logistic regression method is that you have overdose control. So overdose control is the advantages are that it reduces the risks. It exposes patients to drug doses that are below a pre-specified toxicity probability level. I just talked to you about that. See, we discussed about what is called as the target toxicity level. Okay. So now in this one, you set the probability of overdosing, of having a DLT at 25%. So which means that any dose which has a probability of DLT more than 25 is not acceptable to us in our escalation schema. So you always ensure that the dose you are using is less than 25% probability of having a DLT. Okay, and then you will also set the probability of overdosing at less than 25%, which means that it is very unlikely. There is a 75% chance that you are not going to cross the safety dose. And the third thing is, no matter what happens, you will not allow more than 100% dose escalation, which means doubling of the dose. So CRM, as I said, CRM allows you to escalate the dose, like for example, from 100, you may escalate to 500 in CRM, which is almost 400% dose escalation. But the overdose control will never allow you to do that kind of dose escalation. Your escalation will be tightly controlled and it cannot exceed 100. So, and then you are not also allowing the probability of overdosing to exceed 25%, and you are not allowing the probability of DLT to exceed 25%. And because of all these things, you will have a very tight control over the safety aspect of the study, and therefore patients are less likely to undergo overdosing. So some of the important differences between BRRM and 3+3 is the correct MPD, the probability of finding correct MPD, more than 60% of time will accurately estimate the MPD as compared to less than 30% with 3+3. Risk of overdosing is almost non-existent. Risk of underdosing is also not existent because you can at least go up to 100% dose escalation. Fibonacci almost never allows 100% dose escalation. If you remember the Fibonacci sequence, it is like 1.67, 1.5, 1.3. So it is about 60% dose escalation, 50% escalation, 30% dose escalation, but it never allows 100% dose escalation. So risk of underdosing is also curtailed. So risk of overdosing is curtailed, risk of underdosing is curtailed. So that's why this is a very good design. And inclusion of other data in those, like for example, you can put in any kind of data because it uses all kinds of data. So you can have PK data, you can have PD data, you can have biomarker data, you can have efficacy data. Almost everything can be put into this one. The sample size, as for any other Bayesian based method, the sample size is flexible, it is not rigid. But of course, the timelines are very similar for both the 3+3 as well as PLRM. Acceptance by regulatory agencies is also similar. Of course, with the caveat that they would prefer you to spend as little time dosing the patients at subtherapeutic levels. So that is the only caveat that they want to see. And combination dose finding is very feasible in the same study. Once you identify the MTD, if you want to look at the safety of the drug in combination with the drug, in the same study you can answer that question, but that will not be possible with the 3+3 design. So now there are some ethical considerations in phase 1 clinical trials. Patients and their physicians, only those who come to the end of the road, like who do not have any standard lines of treatment, are generally enrolled on a phase 1 clinical trial. And patients with various kinds of advanced malignancies that are, as I said, either refractory to standard treatment or for which no curative treatment exists, are generally enrolled on a phase 1 clinical trial. So that comes to the end of my talk. And just to summarize, we discussed about various rule-based design, like accelerated 3+3 design and the classical 3+3 design and the accelerated iteration design. So these are the rule-based designs we discussed. We also discussed some model-based designs like continuous reassessment method, and then we discussed about BLRM. We discussed about optimal interval, Bayesian optimal interval method, and so on. So I think you have some understanding about these various designs that are being used at this point in time. We also discussed the pros and cons of model-based designs with respect to the standard 3+3, and the ethical considerations, very important, although I dedicated very little time for discussing the ethical considerations, is that the selection of patients are going to choose, and then escalating the dose to the business end, wherein it is longer subtherapeutic. That is very, very important from a phase 1 ethics standpoint. So this point you should never forget when designing a phase 1 clinical trial. You should be always such that you are able to come to the business end of the dose as quickly as possible. So with this, I come to the end of my talk, and I would now like to invite Ms. Chandeli to take over from me for the panel discussion. Thank you very much.
Thank you, Dr. Vikram. Thank you so much, Dr. Mathak. Thank you for your active participation during this entire webinar and nicely covering some interesting aspects of phase 1 studies in oncology therapy area. I would like to take a pause over here. We totally understand that this session has kept us all well engaged throughout, but due to time constraints, we would not be able to take any questions at this point of time. But please be rest assured that we will be sending the answers to each and every question of yours via email. Also, we will be sending across the recorded link of this entire webinar on all the registered email ID. Lastly, I, on behalf of Veeda Clinical Research, would like to sincerely thank you all for being here today and taking out the time to be a part of this wonderful webinar. I would also like to extend my sincere gratitude to Dr. Vikram and Dr. Mathak for being such wonderful presenters and giving us all great learnings today. We at Veeda Clinical Research earnestly look forward to host you all again. Till then, take care and stay safe, everyone.
Thank you very much.
Thank you very much, Yuvragi, and thank you very much, Dr. Vikram. It was a wonderful presentation, and we enjoyed every bit of it. Thank you so much. And I'm sure the audience has benefited quite a lot.
Perhaps we exceeded the time a little bit, but yeah.
That's okay. But it was wonderful. Very nice it was.
Thank you very much.
Thank you. Thank you so much. Thank you, Yuvragi. Bye-bye.
Bye.
Thank you,
Good night.
Yuvragi. Thank you very much, Yuvragi.